So we've set up this construct where we have regulatory elements that all can play a role in the regulation of the nearby gene. Again, remember, these are just sequences of DNA, 6 to 12, 20 base pairs long. So they're very short, that are a specific match for a transcription factor. That's the protein product of another gene that will then bind to it to help regulate this target gene. So we're going to set this up and we're going to add to it and take away from it to show you some of the trends that we see as we evaluate and look at regulatory elements in animals. So it is very easy to have de novo evolution of cis regulatory elements. Because they are only 6 to 12 base pairs long on average, there are a few that are longer, but because they're only that long, it's very common for a random mutation to make a new regulatory element right next to a gene. So much, much more common than de novo evolution of genes. Remember, we talked about where new genes come from? And what we have noticed is that the vast majority of genes can be put into gene families, which are the product of gene duplication events. So although there is some de novo evolution of genes, particularly early in the history of life on Earth and in the origin of some groups, most of the new genes that we see in organisms come about from the duplication of other genes. And there are lots of reasons for this that we won't get into in this lecture. We did that in a previous lecture. It's a little different for regulatory elements. Because a regulatory element is not a very long, complex set of sequences like a gene, right? So genes average about 2,000 base pairs. Some are longer, some are shorter, but about 2,000 base pairs. They need a promoter. On eukaryotes, they have introns and exons, so they're complicated little bits of the genome. And it takes a lot for a series of random mutations to create a new gene, and then have it have any function at all is even less likely. If it does even a very minor function, then it can be taken over by natural selection, and subsequent mutations can be either selected for or against, and it can be shaped by the forces of evolution. But those are very unlikely. Think of it as like winning the lottery. It happens sometimes, but it's incredibly rare. Now, a regulatory element evolving and having some sort of a useful functional effect might be more like, I don't know, winning bingo, right? It's still rare, but happens much more frequently than de novo evolution of genes, okay? So de novo evolution of regulatory elements is a really important thing and happens all the time, okay? So the probability that a region of DNA evolves a protein binding motif, right? So meaning that it's going to bind to some sort of a transcription factor. And then the minimal input for that required for it to be a functional one. That's all that we really need to have it now be useful and then have it be subject to natural selection. So if it's there, if it binds a protein, and if that protein then has some impact on the expression of the gene, boom, you've got the bare minimum. Now, if it's a bad thing, if the organism is expressing a gene in a place it doesn't normally express it, and that makes it less fit, has a negative impact on that organism, it won't be kept, it'll be taken away, and the population will stay the way it was before. If, however, that benefits the organism and it's able to outcompete other members of its population, then it will become more and more common. Subsequent mutations might make it even more beneficial or even more efficient at doing what it's already doing, and then suddenly we have that new feature that becomes a motif or a feature of that population of that species. So it's very common for this to happen when we look at the long term of the evolution of species. And so we can actually calculate a very raw and fairly simple probability. So again, remember I said 6 to 12, here they've got 5 also, 5 to 12. The number of possible random combinations, if you have 5 base pairs in a row, is 1,024. Now this is rather simple math. If you have 1 base pair, there are only 4 combinations, A, T, C, and G. If you have 2 base pairs, then it's 4 times 4 combinations. You have AA, AT, AC, AG, and then all of the other combinations. So there's 16. So with 3, you'd have 64. With 4, you'd have, what's 64 times 4? It's like 254, something like that. And then 256 maybe, anyway. With 5, which is kind of the smallest, there aren't very many of them, but the smallest one means that you're going to have a 1 in 1,000 chance, roughly, of having any 5 base pairs be a specific sequence. So in the Drosophila genome, right, there are approximately 112,000 sites that are going to match this one. So those are potential binding sites for a thing. And the larger our target regulatory element gets, the fewer there are. So there might only be 7. So once we get up to 12, that's very, very specific. Now notice, and there's some corollaries here between primers. Remember when we designed primers? The minimum size for a primer is about 16 base pairs, because once you get to 16, you're going to have a very, very unlikely chance of having those 16 base pairs appear in that order, appear anywhere else in the genome. And once you get up to 20 or 24, which most primers are a little bit larger than 16, then you basically have almost no chance at all of having that sequence anywhere else in the genome. So that's why we design primers that are that long, and it's a waste to design them much longer, because once you hit 16 to 20, they're already so specific that adding more doesn't give you extra specificity. Okay, but notice in humans, which have a larger genome, they're going to be fewer. So by the time we get up to 9, just by chance, there are going to be many fewer of possible binding sites for that target genome. And so just as it is just random chance, probably we have multiple sites in the DNA where these transcription factors are going to bind also, but they're just binding and sticking there, but maybe not having a regulatory impact. It's only when that sequence is in the right position relative to a gene that it's going to have an impact. And then that can be subject to natural selection. Now, you look at this and say, wow, 112,000, how can we even have a regulatory element with 112,000 other random copies, some of which probably don't have any function at all, spread throughout the genome? Well, if that really was the case, it probably would not work, because that protein would be binding all over the place and being sucked up and used in places where it didn't have a function. But an additional feature is that much of the DNA that is not being actively used by cells is bound up in these complexes with histones and other proteins that basically wrap it up and package it away so it's not available for those genes to be expressed. And so there are other control mechanisms that also control regulation at different levels other than just the regulatory elements. Okay, so just make sure you understand that. But for early development and for many other gene expression things, it's really the regulatory elements that are the main thing that we look at, that do 100 to 90 to 100% of the control of the regulation of that gene. Okay, so what is the minimal input for this element to function, and what are the factors that impact that? So let's look at them. Sorry, I don't have them listed here. So let's make a little bit of a list. So the minimal input, what factors are going to affect that? We'll say A, B, C here. So the first thing that can affect it is the length of the regulatory element, right? If it's a short one, then it may have many, many more binding sites, right? And that might do it. So also its specificity. And by specificity, I mean how many proteins are able to bind to that. Is it a single one? Or is it 4, or is it 5, right? And then finally, the main important one is the location, right? Is it right next to a gene so it can control that, or is it not? And so if those things are in the right amount, and we have a new one that pops up from random mutation, we have a protein or set of proteins that are able to bind to it, then suddenly we've got an organism carrying a new genetic feature, a regulation of a gene that was not there before. And so those have occurred across organisms. They've been then subject to natural selection, and the ones that are beneficial and have resulted in a positive outcome for the organisms that carry them have been kept and have often made big major differences, and sometimes even distinguished major groups of organisms. All right, so just review those and make sure you know those. There are others, but those 3 are critical. Okay, the second thing that we've discovered about regulatory elements is that ones that already exist can change and mutate. So here I've just shown it with the same number, but changed its color. So if a mutation occurs in an existing regulatory element, it may no longer bind its original targets, but it might bind another one. Or it might still bind its original target, but maybe a little bit more specifically, or a little more strongly, or a little less strongly. So we can have changes in evolution occurring in regulatory elements. Once they're there, it's not just like it's there or gone. it can be there, it could be changing slightly, so there's a little bit more subtle and more levels of control of regulatory elements than just simply are they there or not. Okay, so we might have a regulatory element that duplicates, and now there are two of them, and that might increase expression levels. We might have the modification that we talked about in the previous slide, right, where it changes and now binds the protein a little less strongly than it did before, and so that has an impact in regulation. And then finally, we may even have cooption. Now, cooption is something that we've talked about previously, and basically what we mean by cooption is a existing gene that is being turned on, and then everything else that it may control can be hijacked, co-opted, that's the technical term. So it can be co-opted in a different set of tissues. So if this blue protein is what activates regulatory element 1 and it turns on the gene, if that blue protein is expressed, a mutation occurs so it's expressed in a different set of cells at the right levels, then we may be turning on this entire gene network. And remember, this is one of the ideas that might have occurred in the evolution of insect wings, right? So one of the early theories was that insect wings had evolved from these gill structures in the ancestors of insects. The other idea was that no, they had evolved from these pronotal extensions, these exoskeleton extensions that we saw on the margins of the thorax. And there was a little bit of evidence from different sources for both of those. A modern theory is that it's actually both together. And in fact, the combined theory, as it's called, might better be called a cooption theory. So it might be that a network to build gills was co-opted and turned on in a location where it was never turned on before, an example of heterotopy. And so we've got these gill-like structures, or at least some of the developmental processes to build gills that are being expressed in a different area with a different genetic background. And so we end up with gills growing on those structures, and they maybe have different features. So they might be sclerotized, right, hardened, instead of soft and able to absorb oxygen. They might look similar but be sclerotized because they are in an area where the genes are also telling those cells to be covered with some sort of a protective cuticle. And so cooption is a feature where we can turn on not just one gene but all of its downstream network targets by simply modifying the regulatory elements that are present in that part of the body. Okay? So, is the evolution of regulatory regions more or less restricted than that of protein coding regions? Well, hopefully, as we've been talking about these, you've gotten the sense that it's much less restrictive. We get duplications that are going to have a minor impact, whereas a duplication of a gene more often has a major impact. We can lose one, or gain one, or modify one, and have subtle changes, so they're a lot more flexible and can change a lot compared to the genes themselves. Okay. Now, transposable elements do play a role in regulatory evolution because transposable elements can copy or insert major chunks of DNA. And so by doing that, we're going to have major impact. And we typically think of those as having an impact on the genes, but they can also have an impact on the regulation of those genes. So transposable elements are just major, or at least have the potential to be major mutations, and they can also be major mutations for regulatory elements. Okay, modifications, right? It's the same concepts apply for protein coding sequences, but there's less rigidity if we're looking at the DNA level. So I just want to make one thing clear. When we're looking at regulatory elements, which are these short little stretches of DNA, there's no translation that occurs, right? It's just a place that binds transcription factors. And so any change at the DNA level is certainly going to have some impact or another on its function as a regulatory element. Where if we're looking at the DNA level of a gene, right, the A's, T's, C's, and G's, there's a lot of flexibility there because of the degeneracy of the DNA code. Meaning that if we change a third position in the reading frame, it's very unlikely that it will have any phenotypic impact. So when I say flexibility of genes to evolve, I mean the amino acid product of those genes are often not very flexible, that they're very conserved. But at the DNA level, we can still have lots and lots of changes that are never carried over to the protein level because they are synonymous changes, right? And there are other forms of changes that have a little impact on the function of the protein also. So there is less in some senses, there's a little bit less rigidity in the evolution of the DNA level of genes, but still a lot of rigidity or a lot of inability for the protein product of that gene to evolve and still have a positive outcome. And so much of that flexibility falls under this class of neutrality, right? That we can get lots of little changes to the third positions and perhaps change the amino acid but not have a positive or a negative impact on the gene. So genes are flexible at the DNA level, but much less at the amino acid level. Whereas regulatory elements really only have the DNA level, right? There are no other levels above that. That's what they are. And so they have more overall flexibility than the genes, but at the DNA level, it's maybe a little bit of a toss-up, right? And we can also have compensatory changes. So if a mutation changes a regulatory element, so it's not quite as good as it was before, but then a second regulatory element occurs de novo, or another one is modified that makes up for that slightly negative thing that maybe wasn't immediately weeded out by natural selection because it was only a slightly negative thing. But if in the meantime a compensatory mutation occurs, we can end back up at the status quo, but have a different complement of regulatory elements because we have changes in one and then compensatory changes nearby or in other ones. And so we can actually have a fair amount of regulatory evolution occurring, changes, mutations, but still have about the same expression patterns. So that again demonstrates the flexibility of these regulatory elements to evolve and still have the same input into the genes that they're controlling. And then cooption, we'll look at this just a little bit because we kind of already just talked about it with wings, but the same concepts apply for protein coding sequences with less rigidity. This applies a little bit here also, but just remember cooption is when we hijack or use an entire network in an area where it wasn't used before, and know that insect wings might be a good example of that. So we can evolve repression of genes, turning off genes, and that may have a very, very different impact than brand new expression, right? So turning off genes is one way to have a downstream end phenotype. So for instance, we turn off distalis in the abdomen, and that results in the ground plan for insects, right, where they don't have limbs in the abdomen. So it's a critical, important morphological change that evolved because of the evolution of repression. We can also have brand new genes, or genes that are expressed in areas where they were never expressed before. So it's not just, oh, let's turn on a new gene and do something new. We could potentially also turn off a gene and its downstream targets, and that could result in something new also. So both repression and expression are critical in the evolution of organisms. And this is just a diagram reminding us that this repressive impact, repression on distalis, that resulted in the ground plan state, the phenotype of the insects.